Alright, Assalamualaikum everyone. How are you all? I hope that you all are doing well. Welcome back to my YouTube channel. In today's video, we are going to be looking at AS Level Biology 9700 topical MCQs of the chapter 2 of AS Level Biology, which is the biological molecules. Alright, so in biological molecules, we have various subtopics like the test for reducing sugars. We are having test for the reducing sugar, the proteins, the lipids, the carbohydrates, fats, right? Basically the test for the biological molecule. This is one of the subtopics of the biological molecule. Then we have the carbohydrates. Similarly, we have the proteins, lipids and water, right? So we are going to be discussing these MCQs one by one. So what we are going to be doing is that in one video we are going to be solving this video in 1, 2, 3, 4 and 5 parts, right? I am going to make 5 videos for the topical MCQs of biological molecules, and my plan is that I am going to cover the in the first video I am going to cover the test for the biological molecules. Like in today's video, I have over here compiled all of the questions related to the test for the biological molecules, right? It covers every aspect of the test of biological molecules. Then in the next video, which would be the part 2, I will be solving the topical MCQs of carbohydrate, and then I would be solving the topical MCQs of proteins, then lipids and then the water, right? So what will happen that after the 5 videos of the topical MCQs of biological molecules, you will have all of the topics covered, right? And you will have a very good grip on the chapter of the biological molecules, right? So let's begin the topical MCQs of biological molecules. Bismillahirrahmanirrahim. So let's begin it. Let's see that what question 1 has to say. Question number 1 is from October-November 2020, paper 1 to question 7. It says that diastase is an enzyme that breaks down starch into maltose. Starch is a polysaccharide. Starch is a polysaccharide, right? And maltose is a disaccharide. Maltose is a disaccharide. Maltose is a reducing sugar, no doubt. A sample of starch is treated with boiled diastase and left for 15 minutes. We know that if an enzyme is boiled, so they are basically denaturing the enzyme, right? Denaturing the enzyme, because they are boiling the enzyme, which means that they are denaturing the enzymes. Sample of the mixture are then tested with the iodine solution. Iodine solution is a test for the starch, test for the presence of the starch, and with the Benedict reagent. Benedict reagent is used for the test of the reducing sugars, right? What are the results? So basically they are over here saying that they are having the starch and they have given the diastase. They have also put the diastase in the solution of a starch, and diastase is an enzyme which breaks down the starch into maltose, which is a reducing sugar. So basically, as they are talking about the boiled diastase, which means that the enzyme is denatured, right? Enzyme is denatured. You need to catch this point because they have written over here that boiled. So you have to catch this point that enzyme is denatured. Because the enzyme is denatured, so the solution of the starch, the solution which contains the starch, will not be broken down, right? Because the enzyme is denatured, so diastase will not work to break down the starch into the maltose, which is a reducing sugar, which means that the starch will be present in the solution and starch would not have been broken down into maltose, which means that reducing sugar will not be present in the solution. So Benedict's reagent will remain blue because the original color for the Benedict's reagent is blue, and then as the reducing sugar is detected, its color changes to green, yellow, orange or red depending upon the intensity of the reducing sugars, right? Iodine solution, we know that iodine solution will turn blue black if the starch is present, and if the starch is not present, it will turn to yellow brown, right? But over here you guys can see that diastase is denatured, so starch will be present, hence the iodine solution will cause the solution to become blue black, right? It will stay blue black. Iodine solution will stay blue black, hence the correct answer over here would be option A. The iodine solution would remain blue black and the Benedict's reagent will remain blue. Acha, alright everyone, also I prepared some of the notes over here for the biological molecules. So let's discuss the test for the starch first with very much detail, so that you guys are able to solve the rest MCQs very easily. So test for the starch. What is the test of the starch? Basically we will add a few drops of the food solution on a white tile. Alright, then we are going to add a few drops of the iodine solution. If the food colors change yellow brown, if the color of the food changes yellow brown, which it means that the starch is absent, and if the color of the food changes blue black, it means that the starch is present, right? Blue black is a positive test for the iodine solution for the starch, basically not the iodine solution, the starch. Basically it is a positive test for the starch, that yes, starch is present. And if it is yellow brown, which means that the starch is not present, it is absent, right? So this is for the starch. Now let's move on to the test for the reducing sugar. Test for the reducing sugar is also very simple. We are going to take a food solution in a test tube. For example, we take it 2 centimeter cubes. Then we are going to add the Benedict solution in the test tube, in the same test tube, for which we have to test the presence of the reducing sugar. So we will take the Benedict solution and the food solution in the same volume. If we are taking the food solution to be 2 centimeter cubes, then we are going to take the Benedict solution also in the to be 2 centimeter cubes, right? Now when we will add the Benedict solution to the food solution in the test tube, so we will observe the color change, right? And if the color changes from blue to red, orange, yellow or green, which I already told you, so it means that the reducing sugar is present. The original color for the Benedict solution is blue, right? And then it will change its color from blue to green, yellow, orange or red if the reducing sugar is present, right? And the color basically depends upon the concentration of the reducing sugar. If the concentration of the reducing sugar is very high, so the color will be red. If it's moderate, it will be orange. If it's low, it would be green, or it would be yellow. And if it's just in a trace amount, just in trace quantity, so it will be green, right? So I hope that the test for the reducing sugars as well as the test for the starch is clear to all of you, right? Now let's move on to question number 2, everyone. Now question number 2 over here is from May June 20 paper 1 question 6. It says that the flow diagram shows the results of a number of tests on a solution of biochemicals. Solution of biochemicals. So Benedict test is blue. If the Benedict test is given to be blue, it means that reducing sugars are not present, right? Not present, because Benedict solution only changes the color if the reducing sugar is present, right? And then it says that non-reducing sugar test yellow. Alright, so if I just do the Benedict test, it comes out to be blue. If I do the non-reducing sugar test, it turns out to be yellow, which means that non-reducing sugars are present. A very famous example of a non-reducing sugar, which is also in our syllabus, is sucrose, right? Which is made up of glucose plus fructose, right? Yeah, glucose plus fructose. Let me check. Yes, it's fructose. Now if I talk about the iodine test is staining blue black, which means that the starch is present over here. Burette test is coming to be purple, which means that the protein is present over here, right? The burette solution basically is blue in color, and if it turns to be purple, so it means that the protein is present. Burette test is basically for the protein, and iodine test is for the starch. Benedict test is for the reducing sugars, and we also have one test for the non-reducing sugar as well. And the very popular example of a non-reducing sugar which is in our syllabus is sucrose, right? Alright, so as you know that the protein is also present, which means that the catalase, which is an enzyme, right? An enzyme is made up of the protein, which means that the protein is also present, right? Glucose is a reducing sugar, and reducing sugar is not present, right? So we will eliminate this option. The other option are also eliminated because it is talking about the lipids, which test is not given to us, and the test and the result both are not given to us in the question. Then if I talk about the sucrose, then yes, sucrose is a non-reducing sugar and it is giving a positive test. Shukar alhamdulillah. So the correct answer for this MCQ would be option D, sucrose, starch and catalase. If I talk about the test for the non-reducing sugar, so what will happen in the non-reducing sugar test, that we are going to boil the solution with the HCl. First, basically, I'll tell you from the start that first we will perform the Benedict test, right? First, we are going to perform the Benedict test. And if the Benedict test does not show any color change, it means that no color change, right? First, we are just simply going to perform the Benedict test for the reducing sugar. And if we observe no color change and the solution remains blue, the original color of the Benedict solution, it means that it is confirmed that there are no reducing sugars present, right? And then what we are going to do, that we are going to boil the solution with the HCl. Why we are boiling the solution with the HCl? In case if there is any non-reducing sugar present, in case that if there is any non-reducing sugar present, for example the sucrose, right? For example, if sucrose is present over there, so if we are going to boil the solution with the HCl, basically we are hydrolyzing it, right? And if we are hydrolyzing it, so sucrose will break down into fructose plus glucose if we are going to hydrolyze and boil it with the HCl, right? So and then we are going to neutralize it with alkali. Why we are going to neutralize it with alkali? Because the Benedict solution work best in the alkaline condition. Work best in the alkaline conditions, right? So we are going to neutralize it with alkali, and then we are going to again repeat the Benedict test. And if now the test is showing any color change, for example yellow, green, orange, it means that the non-reducing sugars were present in the solution. So that's why when we boil the solution with the HCl, the non-reducing sugar broke down into its simpler components, and then we neutralize it, and then we repeat the Benedict test, so it comes out to be positive, right? And the color change is seen. So if the color change is seen after the HCl and then neutralizing, so it means that the non-reducing sugar is present. I hope that this is clear to all of you, right? Now the test for the proteins is left. Let's discuss the test for the proteins as well. So if I discuss the test for the proteins, it is very simple, that for the test of the protein we use the burette reagent, right? And the burette reagent is originally blue in color, right? If the color changes from blue to purple, it means that the proteins are present. Blue color is a positive burette test which indicates the presence of the protein. Negative burette test indicates the absence of the protein, basically, right? So this was a simple test of protein. Now let's move on to question number 3 over here. Question number 3 over here is basically from October-November 2020, paper 1-1, question 7. It says that a student carried out the Benedict test on two different types of the milk, X and Y. A sample of each type of milk was heated to 100 degrees Celsius in a water bath with a Benedict solution, and the time taken for the first appearance of a color change was recorded. The results are shown in the table. Type of the milk X and Y. Time for the appearance of the color change in the Benedict solution 13 and 26. Means that after 13 seconds in the milk X, the color change appeared. The blue color of the Benedict solution turned to any other color from the red, orange, yellow, green, right? And in the type of the milk Y, after 26 seconds, the color change was observed from blue to any color, right? Now they are saying over here that which row shows the biological molecules the student detected in each sample of the milk, and the sample of the milk with the highest concentration of the biological molecule. As the concentration of the as the concentration of the biological molecule would be more, right? So the color change will appear more faster, right? Let's say for example, if the color change is appearing in 13 seconds, it means that the concentration of the reducing sugar is more in the type X milk. Now we know that the reducing sugar which is present in the milk is known as the lactose, right, which is a disaccharide. So it's better that we write the reducing sugar in our answer, that biological molecules present in each sample of the milk. We just take the option for the reducing sugar. We consider the option for the reducing sugar because the lactose is a very commonly said reducing sugar which is present in the milk. No doubt that lactose is a disaccharide and lactose is made up of the glucose and galactose, right? But over here we will consider the reducing sugar as more appropriate, right? Sample of the milk with the highest concentration of the biological molecule. Obviously the solution where the color change was more quick is the solution which is having the highest concentration of the reducing sugar. Hence the correct answer for this MCQ must be option C, reducing sugar and the type of the milk X, right? So hence option C is our correct answer over here. Alright, so everyone, now let's move on to question 4, which is from October-November 2020 paper 1-3 question 7. It says that a student carried out tests for the biological molecules on the same sample of the milk. The test and their results were as follows. Alright, so they have given us the test and the results of the test and the results for the sample of the milk, which they performed on the sample of the milk basically, and then they are saying that which biological molecules must be present in the milk. So we have given the test and their results, and then we have to conclude that which biological molecules are present in the milk. Heating to 80 degrees Celsius with Benedict solution gives the red color. If the Benedict solution is changing the color from blue to red, it means that the reducing sugar, for example lactose, is present, right? Like the reducing sugar which is present in the milk is the lactose. So basically they are saying that the reducing sugar is present, so we can simply take that yes, reducing sugar is present. Adding biuret solution gives the purple color. We know that as the biuret solution changes its color from blue to purple, it means that the proteins are also present in the milk, right? The very popular protein of the milk is the casein. Adding iodine solution gave a yellow color. If the starch was present, then the solution would turn blue-black, but as the iodine solution is giving the yellow color, it means that the starch is not present. So we can simply cross the starch. And if I talk about the non-reducing sugar, so they are saying that boiling with acid followed by the neutralization, then heating to 80 degrees Celsius with the Benedict solution also gave a red color. If the non-reducing sugar, for example sucrose, was present, then what would happen that on the hydrolysis, as we are boiling with the acid, obviously we are hydrolyzing the non-reducing sugar. If the non-reducing sugar was present, then hydrolysis would have broken it down into the reducing sugar, right? Increasing the Benedict's test intensity, right? Intensity of the Benedict test would increase. However, since the test results, you know that the test result remains the same, for example red color, right? Or initially just for the reducing sugar test it was also showing the red color, and for the non-reducing sugar test it is also showing the red color. Since the test results has remained the same, so it indicates the absence of a non-reducing sugar, which it means that non-reducing sugars are not present. If the test result stays the same, it means that the non-reducing sugars are not present. I hope that you guys are getting it. So non-reducing sugars are not present. Only the biological molecules which are present are 2 and 3. Hence the correct answer for this MCQ would be option C. Alright, so now let's move on to question number 5 over here. Question number 5 is from October-November 19 paper 1-1 question 7. It says that which color indicates the lowest concentration of the reducing sugar present in a solution after testing with the Benedict solution. We know that the original color for the Benedict solution is blue in color, and it can give you the color change as red, orange, yellow and green. right? The color depends upon the concentration of the reducing sugar. As we go from green, yellow, orange to red, the concentration of the reducing sugar basically increases, right? So the lowest concentration of the reducing sugar which they are asking us about in the question is indicated by the color green over here. Green indicates the lowest concentration of the reducing sugar and red indicates the highest concentration of the reducing sugar, as we discussed in this test as well, that the highest concentration of the reducing sugar is given by the red color and the lowest concentration of the reducing sugar, which is the trace quantities of the reducing sugar, it is given by the green color, right? So hence the correct answer for this MCQ must be option B, which is green. Alright, so everyone, now let's move on to question number 6 over here. Question number 6 is from May June 2020, paper 1 to question 7. The question over here says that a student carried out the Benedict test on a sample and got a negative result. What should the student do to confirm there are no sugars present in the sample? Right? Alright, so basically if you understand the question over here, so they say that in the Benedict test, the student got the negative result, which indicates that there is no reducing sugar present. However, there must be non-reducing sugars present, right? So we need to confirm that no reducing sugars are also present in the sample. So to confirm this, we have to do the non-reducing sugar test, which is that we are going to boil the solution with the hydrochloric acid, right? Why we will boil it with the hydrochloric acid? To hydrolyze the non-reducing sugar, for example the non-reducing sugar sucrose. In case if only sucrose is present, so it should be broken down into fructose plus glucose if it is boiled with the hydrochloric acid. Then what we have to do is that we have to neutralize it with the alkali. Why? Because the Benedict solution or the Benedict reagent works the best in the alkaline condition. And then we are going to repeat the Benedict test. If no color change is seen, if after the Benedict test the solution remains blue, it indicates that the reducing sugars are also not present, right? So hence the correct answer for this MCQ must be option C, that only after doing the non-reducing sugar test we can confirm that no sugars are present in the sample. From the Benedict test we confirm that no reducing sugars are present, and from the non-reducing sugar test we confirm that no non-reducing sugar is either present in the solution, right? So we can say that no sugar is present in the solution, then only we can say that, right? So I hope that you got the question number 6. Now let's move on to question number 7. Alright, so now let's end this video over here. We will continue from the next question in the next class. Have a nice day everyone. Take care. Allah Hafiz.